have cells in this organization. We're complex. We're sensitive to the environment. Like we in this room right now, it's cold, and we're cold, so we're responsive to the environment. We grow, we reproduce, we absolutely use energy. So, we adapt to we adapt evolutionarily, okay? Now, here's what I would suggest. I would suggest that instead of you writing a whole bunch, that if you have your laptops and you have because this is all in there, and that way you don't have to spend a lot of time, not unless you write in order to help you memorize, because sometimes people like to write to help themselves memorize things. But this is all in there, and we're gonna go through this because we have 3 chapters to cover in 2 hours. So, let me see something. I was gonna do a virtual on this, but I just don't. Let me see this courtroom. See if there's anybody in that room. Every once in a while, I'll check. If there's nobody in it, I'm not gonna go there. There's nobody in it. Okay, so I'm gonna close it. Okay, all right. So now there are certain. Here's where it's gonna get where I really want you to focus to understand. You don't have to go through all of this. I'm not gonna even cover this. When it comes to, I'm not gonna cover any of this as well. I'm just gonna tell you straight off. This is scientific approaches. I want you to take a look at it on your own, but it's not gonna be on the exam. So we'll start with Darwin's Origin of the Species. Understand what it means by natural selection. Darwin actually proposed this whole theory of how he theorized that evolution occurs because of something that happened in the environment, and because, like for instance, a particular bird no longer had a certain source of eating, didn't have the same eating, bugs disappeared or something. And that's what he called natural selection. It was selection based on nature, number one, and also it was survival of the fittest. So nature selected for who was going to live. And so the environment, it was a natural selection, environmentally influenced. That's maybe the point that I want to make. Okay, and that's what this natural selection means. It means nature selected for this process. Okay, I want you to understand that the evolutionists believe that, and here it is again, the people that believe in evolution, they believe that there is survival of the fittest, meaning only the fittest survive to reproduce. And that this survival is based on something that happened in the environment. So it was naturally selected. It was selected by nature. Okay. And the thing that I want you to understand is, I wish I had a pen. Things that I want you to understand is that the evolutionary process is a genetic change. It's not a change that happens. It's a gene-based change. Thank you. Mm-hmm. That's an extra one you have. Can I have a piece of paper? Mm-hmm. Yeah. It's a gene-based change. It's genetic. It doesn't just happen because, are you with me on that? It's a gene-based change, which means that it can be passed on to the offspring. All right? So it's a gene-based change. Okay. All right. Okay, all right. Okay, all right. So understand that. And I also want you to know this term about descendants with modification. Okay, okay? What that means is that it means that the offspring, whoever has evolved, the organism that has evolved, meaning the offspring, it means that they look like their descendants, but they have changes that make them look like their ancestors. So descendants with modifications. So they have genetic modifications. That's what that means. It means that whoever has evolved, whatever organism has evolved, they're gonna look like their descendants. They're gonna look like their ancestors. The descendants are gonna look like their ancestors, but they're gonna have some modifications, some changes. That's all that means. And those changes are genetic, otherwise. So I want you to understand that term, descendants with modification. And actually, he put that in his text. He talks about natural selection, survival of the fittest, and descendants with modifications. And all it means is that the offspring, they're the descendants, they look like their ancestors, but they have modifications. Are you with me? Mm-hmm. They have changes. That's what modifications mean. So all it means is genetically, they look like their ancestors. They look like the ancestors, like you look like your grandma, or your third degree or whatever. But genetically, you're modified, you're changed. That's all that means. And it happens with evolution as well. Okay. All right, now you don't have to know any of that, all of that. That's the point. And all of this, I want you to understand this, but quite frankly, it's not going to be on the exam. But this is just explaining, Darwin had these theories. His theories were based on looking at what we call the physical characteristics of something. And he compared the current day organisms to the fossil organisms just from comparing what the physical appearance. Fossils are the old ones, the ones that are, fossils that are crushed between the rocks. Fossils, right? Mm-hmm. Yes or no? Fossils? Yes. Yeah, the fossils. Okay. And so he compared the fossils, which are crushed between the rocks, to the birds and all the other organisms he was looking at while he was on the Galapagos Islands. And he was saying, okay, these look, and he was basing all of his theories from that Origin of the Species book was based on looking at physical characteristics. And all these later slides say is that Darwin was right, because now we've looked at the genetics, the genes of the fossils compared to the present day ones, and we said, okay, we validated what Darwin said and thought. Are you with me on that? So that's all that is. Now I'm not going to go through all of this because this is just extra stuff. Okay, now, we'll talk about that later. I want you to know the difference between DNA and RNA. Okay, this latter part, I want you to know the difference between DNA and RNA. DNA, deoxyribonucleic acid. And, it's made up of adenine, thymine, guanine, and cytosine. I'm not sure if this is on here, but I'm gonna just mention this is a karyotype. This is all your chromosomes. I probably won't have this on the exam, but this is what a gene looks like. I do want you to know that for DNA, and let me get to that slide. DNA is, where is that? No, it's not on here. It's on here somewhere. I think that I went over, because I asked you this in class, the difference between DNA and RNA. DNA and RNA, I'm just trying to get to the slide, and maybe it's not on here. But RNA is a single-stranded molecule. DNA is double-stranded. And I think it should be in one of these slides, and I don't see it actually. We went over this in class, right? Mm-hmm. Yeah. Now here's something that I want you to know. It's not on this set of slides, but it should be on here. So let me go to review. Okay, all right, I want you to know that I'm gonna draw a structure. Yeah, I don't know if this is correct or not. It's got CH. Oh no, this is, hold on one second. Okay. DNA has, hold on, let me get my pen back. Okay, DNA has a phosphate group right here. It has a phosphate group right here. And then it has a nitrogenous base right here. Nitrogenous base, okay? Okay, so I'm trying to, DNA is made up of adenine, thymine, guanine, cytosines. It has a nitrogenous base, it has a purine. Let me see something. Hold on one second. Okay, this way I don't have to write it. Okay, I don't have to draw it, I can have it right here. So this is just one of the bases that make up DNA. Here's what I want you to know about DNA compared to RNA. For DNA, at this number 2 position right here, I might have this as an extra credit point actually. At this number 2 position, did I go over this in class? At this number 2 position right here, DNA has an has a hydrogen, and an RNA has an OH. They call it DNA because it's D, meaning without oxy. Are you with me on that? So that's where DNA gets its name. So if I have right here on this portion right here, on this carbon right here, this carbon where I have this point, for DNA, it's just the H. And they call that deoxy. D meaning without an oxy. And for RNA, that's how it gets its name. For RNA, it has an OH right here. So instead of right here, it has an OH like this. So RNA has an OH right here. DNA has an H. Are you with me? I think I'm gonna make that as an extra credit point. Okay? So are you with me on that? So that's how DNA got its name. It's D meaning without oxy. Deoxyribonucleic acid. And RNA is ribonucleic acid. Okay? Are you with me on that? Okay. All right. So anyway, and of course, actually, that's in chapter 3. We talked about that in chapter 3. Okay? So it is fair game. I don't have to have it as an extra credit. I can have it as a real point because I talked about that in class. That's in chapter 3, but I'll talk about it. That's how DNA got its name. Are you with me? Deoxy, meaning without an oxy. Right there. RNA has an OH right, see that OH right there? It has an RNA, both of them have an OH right here, but RNA has an OH right here and right here. And DNA just has an H right there. Hence the name deoxy. D means without an oxy, okay? Without an oxy, without an oxygen. and it's got that, okay? Are you with me? All right. All right, so let's go back to this. And we'll talk more about DNA once we get to chapter 3, because in chapter 3, we talk about linking a bunch of molecules together to make a longer molecule. Remember, that's what chapter 3 is about. So that's where I actually talk about all this stuff. Okay. Are there any questions? Chapter 1 is going to be, I'm not going to get a lot from chapter 1. Excuse me. Most of it is, but it's going to be what we've talked about. Yeah, okay. All right, so let's go. Perry, can you start a roll for me? here's what I normally do. I don't tell people about it, but people that come to the review, at the end of the semester, you kind of get a little curve, an extra curve if you need it. Okay, if you get an extra curve. Okay. All right. So anyway, let's start on chapter. Why do I have all these? Oh, this is chapter 1. Oh, so let's go with chapter 2. Now, chapter 2, I'm going to explain some things, and I want you to, okay, I want you to know the difference between, all of the subatomic particles. The subatomic means, sub means below atom. An atom is an element, sodium, chloride, sodium, all that stuff. Sub meaning less than, what makes that up. So that would be the protons, which are positive, the neutrons, which are negative, the electron, the neutral, the electrons, which are negative. I want you to know that the particular element, and the electrons rotate, of course. The particular elements are the things you see on the periodic table. Sodium, chloride, hydrogen, oxygen, all those things. Those are elements. The number of protons will determine what that element is. Are you with me? If the number of protons, not the number of neutrons, not the number of electrons, the number of protons will determine. So if you look on the periodic table, so I have an example of the periodic table, the number of protons will determine what that is. So every time I see 1, I'm hydrogen. Every time I see 8, is it 8 or 6? It's oxygen. Every time I see 6, it's carbon. Are you with me? So the number of protons will determine what that is. So it's the proton number. The proton number, and that they call that the atomic number. Don't be confused, but it's the atomic number. Okay. Now you can change the neutron number. The neutron number, if you change the neutron number of something, it becomes an isotope. Iso meaning equal. And let me show you what that is. Okay, so let me show you the isotope. So for the isotope, so see right here, here's the example where I have carbon. I have carbon, this is carbon, this is a carbon, this is a different kind of carbon, this is a different kind of carbon, but they're all carbon. But notice they have the same proton number. See this proton number right here? Proton 6, this is carbon 12, carbon 13, carbon 14. These are radioactive, by the way. Okay, they can be found in the hills of Montana. I say that all the time because that's where they can be found. But they're all carbon. It's a carbon, this is a carbon, and this is a carbon. What varies is the number of neutrons, which are these neutral colored ones here. The neutron number is different. Those become radioactive isotopes. You know about radioactivity. It becomes a radioactive isotope. So it's an iso meaning equal, but don't get that confused with the proton number. The proton number determines it's a carbon. Proton number, it makes it a carbon. Proton number makes it a carbon. And you can actually see that on the periodic table. The neutron number will change for a radioactive isotope, actually. Okay? And those are important because they are radioactive. And you know what radioactivity does. Okay? It obliterates stuff. So note the proton number determines what element it is. If I change this proton number to something else, it's going to be a different element. It's not going to be carbon anymore. That's what I want you to know. Right? But the neutron number can change, and when the neutron it becomes a radioactive isotope. Okay. All right. And it has all the qualities of radioactivity, actually. Okay, now I do want you to know about this octet rule. This octet rule, octet means 8. In order for an element, now here's where it becomes really important. I'm not sure I was able to have you guys understand this in class. The octet rule is really important because it determines who that element is available, who's that element can interact with. The element, if an element has, and I'm gonna ask you something like this, it wants to, as long as it has 8 electrons in the outermost shell, 2, 4, 6, 8, like neon, it's a happy, stable element. It doesn't need to interact with anybody. Are you with me? It's stable once this outermost shell is occupied with up to 8 electrons. So if I have an element that has, and that's how these interactions happen in your body. If I have an element that has 5 electrons, and I'm gonna ask you something like this. If I have an electron that has like 5 electrons in the outermost shell, who does it want to interact with in order to make itself stable molecule? Somebody who has how many electrons? 5 plus 3 is 8. Are you with me? That's how these interactions, that's how element to element interactions are determined. They don't just randomly interact in your body. They're interact. Are you with me on that? So if I have something that has 4 electrons in the outermost shell, who do you think is gonna want to interact with? Someone who has how many electrons? 4. Oh, 4. 4. I said 4. 4. Are you with me? That's how these interactions are determined. They don't just randomly knock up to each other and make them, because when I say interaction, I mean making a big molecule. Because one molecule binds to another molecule, binds to another molecule, like to make a whole big carbon. Are you with me on that? So those interactions are based on satisfying the electrons in the outermost shell. Okay? Satisfying this. And it's called an octet rule. I don't know if it's on here, but it's called an octet rule. It might be on the other slide. It's on here somewhere. Should be octet rule. Okay, energy level. Are you with me? Are you with me on that? Because I'm going to ask you something like that. All right. Where is that octet rule? Okay, here it is right here. Here's the octet rule. That means 8. Octet. Octet means 8. So if I have 2 electrons, I want 6. I want to couple with somebody who's going to give me 6 electrons. Okay, so that I can be a stable molecule. Now that neon, it's very happy. It does not interact with anybody. Neon is a gas. And it's got 2, 4, 6, 8 electrons in the outermost. It doesn't need to interact with anybody, right? So it's very happy. Okay, now I skipped over some things. I want to go back to those because I'm going to take a little bit more from this chapter, from 2 and 3 than from 1. Because we skipped over a whole bunch of stuff in 1. Okay, so let's see. Electrons in the outermost shell are much more energetic. And actually that becomes very important. That becomes very important when we start talking about, in later chapters, when we start talking about how you generate energy from your food. So I want you to remember that. The electrons in the outermost shell are the most energetic. They have the most energy. According to the physicists, they have the most energy. I absolutely want you to know oxidation reduction. Absolutely. I want you to know what oxidation is, what reduction is. Okay? Now, we're talking about, for oxidation is an element that loses electrons. Reduction, now electrons are negative. Remember, electrons are negative. Reduction is an element that gains electrons. So here's the electron that's going from this element to this element. And here's what I said in class. I said the way that I used to remember this when I was in high school was that for reduction, electrons are negative. Whenever you're receiving something negative, you become less or reduced. So, and the other one is oxidation, okay? So figure out how you want to understand this, but I want you to know the difference between oxidation and reduction, okay? I want you to know the difference between oxidation. So the question is going to be pretty straightforward, but just know, if you gain an electron, if you accept an electron, like here, the electron is going from this element to this element. Because it's negative, electrons are negative, you're accepting something that is negative, you become reduced. That is going to become very important when we start talking about the process by which you generate energy from your food. That's going to become critical. Oxidation reduction, that term is going to become important. And also this idea of the energy on the outermost shell. Not right now, but later. So I want you to put it in your brain cells, okay? All right. Okay, here's the periodic table. You don't really have to know this, you really can't see it, but that number at the top, this is a bad screenshot, but that's just the proton number. And the proton number determines what element it is. The proton number determines the element. Okay, all right. So every time you see 6, you don't have to know that, but every time you see that, I'm telling you, every time you see 6, it's a carbon. Every time 1, hydrogen. 8, oxygen. So, but just know that the proton number determines what it is. They call that the atomic number, the atom number, the atomic number. Okay. Now, y'all gonna have to stop me if you want me to kind of back up a little bit. Oh, absolutely know what valence electrons are the electrons in the outermost shell. That's what the valence ones are. And quite frankly, the valence ones are, when we start talking about how you generate energy from your food, we're gonna just use the term valence electrons. Valence electrons in the outermost shell. That's what valence ones are, okay? All right, and I say right here, here oxygen, for water, oxygen has 6 electrons in its outermost shell, and it interacts with how many hydrogens? Exactly, okay? That's how these interactions are determined. That's how these bonding interactions are determined. They don't just bond together. Okay, now, I have this one in here. This is the little cheat sheet I have in here. Should be in yours as well. 2 needs 6 to make, 2 on the outermost shell for magnesium. It needs 6 to make it a stable molecule. And the same thing, 6 gonna need 2. This is oxygen, it has 6, and then binds 2. Anybody with 2 electrons. Okay. Okay, now. That's what we talked about before. Now, absolutely, I want you to know carbon, oxygen, hydrogen, nitrogen. I talked about that all the time. Carbon, oxygen, hydrogen, nitrogen. It makes up 96% of who you are. 96%. And you look at anything that's in your body, your carbs, your proteins, your lipids, what else did I miss? Carbs, proteins, lipids, nucleic acid. If you look at the structure, what makes up all of those molecules, you're gonna find carbon, oxygen, hydrogen, nitrogen. Those are the 4. Okay? So just know those. Find out a way to understand that. And there are also what we call trace elements. And actually, this is another, yeah, you don't have to know that. Okay, this is another. Now what I have is, hold on 1 second. Okay, so what I have here is a nucleotide, a nitrogenous base. And I use this in class as my example of the importance of carbon, oxygen, hydrogen, nitrogen. And I told you that even if you look at, that's thymine. Remember, DNA is made up of adenine, thymine, guanine, cytosine. Remember? Adenine, thymine, your genes. And I used this as an example, and I said, you can find carbon, oxygen, hydrogen, nitrogen in one of those nucleotides. That's the point I want to emphasize for it. That's why these things are so important. Carbon, oxygen, hydrogen, nitrogen. I have a nitrogen, I have a carbon, I have an oxygen, I have a hydrogen, and I have nitrogen. So it's got all four of those. So all four of those are in all of my DNA. And the same thing can be true of lipids. They're going to include carbon, oxygen, everything except nitrogen. Do you see the point? The point is that's why those four elements are so important. They're used to make up your protein. Your proteins have carbon, oxygen, hydrogen, nitrogen. Your amino acids. Do you see the point? So that's why they're so important. Okay? They're in everything we have. And then I have this as an example here as trace elements. So I say that trace elements, people always get that confused. They say that trace elements are, they're not important. That's not true. It means you just need a trace amount of them. You need just a little bit, like vitamins. And I have as an example, iron that's in your blood. Iron that binds to your hemoglobin, that's a trace element. You only need a little bit of it, but if you don't have it, you're not going to be able to carry oxygen to your tissues. Okay? But the point is, you just need trace amounts. That's all that means. But they are critically important. So don't think that trace elements are not important. They're very important. And I had my example, iodine, because iodine is one of the trace elements. Here's iodine right here. And I told you that I asked you in class, I said, when you go home and you look at your salt, the salt that you buy from HEB, the salt sodium chloride that you buy from HEB, look on the box. It's going to say iodinated or non-iodinated, because now the government has made it so that companies will tell you whether or not there's iodine supplemented in your salt or not. Iodine has been added because if you don't have it, it can be bad. I was going to say catastrophic. It can be bad, actually. I'm not going to go into what happens, but it can be bad. But that's just to emphasize the importance of the trace elements. You just need trace amounts. They're still very important. Okay. All right. So know that. Now let's go to the bonding interactions. For the bonding interactions, the whole purpose of that is we're starting with atoms. We're starting with, atoms, subatomic particles, neutrons, protons, all of that. And then we're going to elements, which are the oxygen, nitrogen, hydrogen, all of those. And now we're going to start building things together. By building things together, we're going to start linking individual molecules together to make a big molecule, to make some of the stuff that makes up your body. So I said there were four different kinds of bonding interactions, linking interactions. There's covalent, ionic, hydrogen bonding interaction, and I included Van der Waals forces. And I just named them in the order of strength. Covalent is the strongest, ionic is the next, hydrogen bonding interaction is the next, and Van der Waals is the next, okay, the strongest. And here's what happens with, so I hope you can see, for covalent, this is where you see double bonds. You've seen double bonds associated with molecules. Ionic, covalent bonding is where, and I use this in class, I say electrons are moving. According to the physicists, electrons are orbiting. They're moving. They're not at one particular place. They're rotating like this. And for covalent, so this is a carbon, this is one oxygen, this is another oxygen. For covalent, the orbitals are overlapping, which means that they share space. Are you with me on here? There can be orbiting like this if they're not bound. So this is, did you get what I'm saying? Yes or no?
Yes.
Okay, let's say that I have two elements, I have two independent elements. like a sodium and a chloride. And the electrons are always orbiting around the nucleus of that particular element. According to the physicists, the electrons are not in one place, they're orbiting. But in the case of, for sodium chloride, the sodium's gonna be out here, the electrons are far removed from each other. The orbiting electrons are far removed from each other. For the covalent linkages, they're close to each other, so close that they share electrons. So even if I have one electron here and I have one electron here, because they're orbiting so close to each other, they're sharing electrons. They're sharing the properties associated with them. Are you with me on that? So that's what that sharing is about. Are you with me? So and I draw my hand like this because they're moving and they're actually sharing electrons. So it's not, if I have one here and one here, I really have more than one because I'm sharing the other one that's orbiting with me. Okay? So that's what happens during covalent bond. That's why they're so tight, because it's not just one electron here and one electron here. It's got two electrons because it's sharing electrons that are orbiting close to it. Are you with me? That makes it very strong. And I think in class, I use the example, you might have been looking down, I use in class, it's like a chain link fence. You know how the chain link fence is one piece is linked to the other piece like that, and it makes it real strong. Okay? And a double is like this, and a triple is like this. Okay? And that's exactly what happens, because I'm sharing, this is sharing with this. Are you with me? Now, and the other ones are removed like this. They're not sharing, they're just isolated. Okay? So here's what I want you to know. I want you to know covalent is strongest, and then next is ionic, is for a strength. Next is hydrogen bonding. Next is van der Waals. Now I'm not sure whether van der Waals is really not in your text, but I do want you to understand that there is a fourth bonding interaction. Now, okay? Now, is this too much? Okay. Now here's what I want you to understand about, I want you to know the difference between polar and nonpolar. Nonpolar and polar. Water is polar, and polar means hydrophilic, water loving. Okay? And I want you to know the difference between hydrophilic and hydrophobic. Okay, hydrophobic and hydrophilic. Okay, now there was something. Oh, here's the ionic. Now I'm gonna go through these really fast because I want to spend a lot more time on chapter 3, because I think I need to, but it's gonna be about the same amount of information that I get from 3 and 2. But judging from the looks on y'all's faces in class, I think I need to spend a little time on 3. Okay, now here's what I want you to understand about. So the next one is ionic. The best example that everybody uses for ionic is sodium chloride. Everybody uses that. That's the best example. Because here's what happens. This is a little fuzzy. Now, this is, okay, so here's sodium, and here's chloride, which is regular table salt. Okay, I want you to know that during this bonding interaction, and I'm going to explain it. You don't have to know all this, but I'm going to explain it to you so you can understand it. This is an oxidation. The sodium becomes oxidized because it loses an electron. The chlorine becomes reduced because it gains that electron. We're back to oxidation reduction again. So, all right. Now, because this loses an electron and this gains an electron, because first let's go because this loses. This loses an electron. It has one electron in its outermost shell that's going to be captured by this chlorine. Because it loses something negative, it becomes overall positive. And because this gains something negative and it's reduced, it becomes negative. So this becomes positive, this becomes negative. For the ionic interaction, and I mentioned this in the class and I'm gonna mention it again. For all the interactions except for covalent, they are opposites attract. Remember me saying that? All of these are opposites attract. Positive attracting to a negative. You've heard that term, opposites attract? They're opposites attract. So that's the bonding interaction between, for your table salt, the thing that keeps the sodium chloride together is one's positive, one's negative, and their opposites attract. And that's the same for everything, all of the bonding interactions, except for covalent. Ionic, hydrogen bonding interactions, opposites attract. It's overall positive, negative interaction. Okay. Van der Waals, overall positive, negative interaction. Okay. Okay. So this is a bigger picture of it. It becomes, so we can cover a lot of things. We can call it oxidation reduction right here, because this becomes, it's losing something, it becomes oxidized, it gains something negative, it becomes reduced or less. Reduce means less, okay? And overall, it's opposites attract. Positive interacting with the negative. Positive interacting with the negative. So opposites attract is what's keeping that sodium chloride together, okay? So what I want you to know about that, I do want you to know that it's not as strong as the covalent linkage, because it's next on the list. Covalent, ionic, hydrogen bonding interaction is next, and then Van der Waals is the next. Now, you'll notice that this is water, and see, this is also opposite attract, because oxygen is negative, hydrogen is positive. So here again, we have an opposites attract, but it's not as strong as the ionic and the covalent. So I keep mentioning that because that must mean you're going to see it. Covalent, ionic, hydrogen bonding interaction, and Van der Waals in that order. In that order, okay? And here you have opposites attract again. You can have it again. So here's and this is called hydrogen. This is hydrogen bonding. It involves a lot of hydrogen bonding, a lot of hydrogens. So here's the positive from the hydrogen, the negative from the oxygen. Okay? All right. Okay, now, let's see, what do we have? Let me see. I want you to know these terms, cohesion and adhesion. Now, cohesion and adhesion. Okay, cohesion, the cohesion is water molecule interacting with another water molecule. Miss Carey has some water on her desk. So that is really, water interactions. Millions of water molecules interacting with millions of water molecules. That's what cohesion is. Adhesion is water with something else. If I throw some salt into her water, then the sodium chloride, the positive associated with the sodium is gonna bind to the negative of the oxygen. And the negative of the chloride is gonna bind with the positive of the hydrogen. Okay, oops. Okay, so, and that's what this is. That's what this is demonstrating. And so this would be an adhesion interaction, because it's water with something else. Water with sodium chloride, okay? Water with sodium chloride. Okay, I think that, I'm not sure about the properties of water. I'll have to think about this. Properties of water. I do want to mention that every single property of water is based on its ability to form a hydrogen bond interaction, meaning a positive negative interaction. Okay, that's probably all I want you to know about that.
Its ability to form what?
Say it again.
Its ability to form
to hydrogen bond, to interact with the H2O. Okay? Now, I want you to know what pH is, what the pH range is. I want you to know what a buffer is. A buffer is a weak acid and a weak base. A buffer is a solution that's going to keep your body from changing pH, to make drastic pH changes. And that's because it's a weak acid and a weak base, which allows it to dissociate really quickly. It can get rid of hydrogen ion. It can get rid of hydrogens real fast. Attach the interaction. It can bind hydrogens really quickly and release them very quickly. That's what all that amounts to. But I want you to know that buffers are, by definition, are weak acid and weak bases, which means that they can interact with things and interact with whoever they're going to interact with fast, and they can receive and give up those interactions very quickly, okay, which makes them weak. And the acidic pH, is in the range of 0 to 6, and it's a high hydrogen ion concentration, high hydrogen ion concentration. And of course, basic or alkaline is a low hydrogen ion concentration. So, and this is just an example. The most acidic portion of your body is your stomach, of course. Your stomach, which is like a pH of 2, 3. Okay, this is the weak acid and the weak base. You don't have to know the chemical structure, but that's what it looks like. So that's all you have to know is that it's a weak acid and a weak base. Okay, and by weak it means that it can bind. If there's I'll just give you an example. If your body, for some reason, you know, your stomach is got acid reflux or something. Acid reflux is where the acid in your stomach is not confined to your stomach. There are valves at the top of your stomach. They're stoppers. We call them valves at the top of your stomach and the bottom of your stomach that open and close when you get ready to eat. And there are some cases where those valves don't work properly, and so it might stay open. So the acid from your stomach is going to rise up into your esophagus, and they call that acid reflux. And some people have it because the acid in their stomach is just coming up into their throat, into their esophagus, because the valve is open. It's supposed to be closed, it's open, and the acid comes up. And for some reason, if you know you have a high concentration of acid in your body, and it's not, in your esophagus, you use your mouth, your esophagus, and then your stomach. For those of you that haven't taken anatomy yet, yeah, it's your mouth. Food goes in, it goes down your esophagus, which is a hole, and then the valve in your stomach opens up, it goes in there, and then it's supposed to close like that. But acid reflux, it stays open and the acid comes up. Acid is high hydrogen ion concentration. It means that's the definition of acid. A lot of hydrogen ions. Hydrogen ions are extremely acidic. And if you have that, then there is this molecule, this molecule right here, the molecule that can actually bind to that hydrogen and take it out of solution. Are you with me on that? Long story short, it captures the hydrogen, captures the acid. And then it can release the acid, it can capture and release very quickly. Okay? So that's all that is. Okay. All right, so let's go on to chapter 3 and spend a little bit of time.